Good evening and welcome to the Dover Lyme and Tick-Borne Disease Forum organized by the Dover Board of Health Lyme Disease Committee. I am Barbara Rothschechter. I'm presently the president of the Dover Board of Health, and I am the chair of the Lyme Disease Committee. Before we get started, allow me to introduce the members of the Lyme Disease Committee and of the Board of Health. If you could stand up and turn around. Here in the front row is Mary Ablé, Angela Wilson-Taylor, Tim Holliner, Paul Tedesco, Steve Kruskal, Matthew Schmidt, and Jim Palmer. Thank you. The members of the Board of Health that managed to come here, there is Dr. Joe Musto, and there is Diane Fielding, who is the administrative assistant of the Board of Health. Most of these members have been working with the Lyme Disease Committee for at least 5 years. And Diane Fielding, the administrative assistant of the Board of Health, has been with us for over 10 years. Some of us will be speaking to you at the end of the official meeting. We will do this in an unofficial manner in the back of the room when every formal presentation is over. Otherwise, we will not have enough time for our outstanding speakers to present their data. So, over the last 5 years, under the supervision of the Dover Board of Health, the mission of the Lyme Disease Committee has been to seek the reduction of Lyme disease and other tick-borne diseases in Dover and surrounding towns by its three-pronged approach. Number one, extensive and continuous education of residents about the means of personal protection. Number two, continuous and extensive education of residents and organizations about the means of property protection by creating tick-safe zones on private and recreational land. And number three, management of the deer population via a responsible deer management plan. Based on these principles, tonight's program consists of the following segments. Why is this not working? We had it before. Can you lift up the screen? Tim, could you have a look, please? This is, I'm terribly sorry. Here we go. Perfect. Thank you very much. There are 4 segments to our forum tonight. The first segment will be a presentation of the Board of Health Lyme Disease Committee activities that were conducted between 2012 and the beginning of this year. In particular, we have 2 very interesting research projects that were conducted during the summer of 2012, and you all are aware of our Lyme disease survey that was conducted during the beginning of this year. Thereafter, Dr. Sam Telford will talk about ticks, ecology and management, and I will introduce both Dr. Telford and Dr. Sam Donta when we get to their specific segments. But Dr. Donta will talk about Lyme and other tick-borne diseases, symptoms, and treatment options. And the last segment is a question and answer period. I would like to take a minute to explain the logistics of the question and answer period. You have seen all the index cards and the pens in the back that were handed to you. Please feel free to write down your questions during the presentations of our guest speakers or after, and then hand them over to members of the Lyme Disease Committees who will come around and collect your questions. The green cards are for Dr. Telford, and the yellow cards are for Dr. Donta. So, let me begin by summarizing for you 2 of our local research projects. Both projects were executed by an outstanding DPH, Department of Public Health, intern by the name of Megan Burns. Unfortunately, she couldn't make it tonight, but I want to give her the credit she deserves. The first project addressed the obligatory reporting of a Lyme disease diagnosis in Massachusetts. You may not be aware of this, but Lyme disease is considered a reportable disease, which means that any physician who makes that diagnosis has to report it to the DPH office in writing. And there are both federal and local agencies that believe that the official reporting that is being done about Lyme, the diagnoses of Lyme disease, are actually underreported. There are very good reasons for this assumption. Therefore, in our first project, we actually send out an anonymous survey to physicians in 4 hospitals in the Boston Metro West area and asking them whether they had any diagnosis of Lyme disease during the past year, and if so, how many, whether they reported them, and if they did not report them, why not? And these results are on the next slide. Tim. Why don't you sit right next to it, please? There we go. A total of 472 physicians received the anonymous survey, and 180 of them responded. That amounts to a 38% response rate. That is pretty good. These 180 physicians made 2,670 diagnoses of Lyme disease during the past year. Of the responding physicians, of the 180 responding physicians, 42% indicated that they had reported all the diagnoses they had made. 32% that they had reported some of the diagnoses, and 24% indicated that they did not report any of the Lyme disease diagnoses they had made. As for the reasons for not reporting, they indicated that the reporting process was too cumbersome, 47%, and it is indeed a big and extensive reporting form that has to be filled out by the physician, and that Lyme disease was too common. From these results, we then drew the following conclusion. Could one of you guys sit close by, please? Thank you, yes. The conclusions then is that Lyme disease is indeed underreported, and agencies may be right in saying that this is the silent epidemic that we still have to identify and then manage and treat. The data reflect a small sample of special physicians because these were hospital physicians. And it might very well be that the respondents were biased in the sense that those people that reported the disease reliably actually were more responsive to our anonymous survey and answered them. So it could be that the data are artificially high, but we do not know this, but we do have the numbers that I reported to you. In order to help the communities and the entire country, it seems to be obvious it would help if the reporting process could be simplified. And last but not least, some of the physicians might not even be aware of the fact that Lyme disease is a reportable disease. The purpose of our second program was to seek to decrease the incidence of Lyme disease by educating Dover residents about tick habitat. Tick population densities in local outdoor recreation sites were measured to provide additional tick safety recommendation if necessary. Ticks were collected once weekly for 5 consecutive weeks using a sampling technique developed by Dr. Telford and validated by Dr. Telford. And they were collected at 5 recreational sites in Dover, each having a sunny and a shady transect. They were collected always at the same time of day, anytime between 10 and 1 p.m., not necessarily on the same day, because that was determined by the weather. I could not collect ticks when it's raining. The next slide shows us the result. I think it is a pretty self-explanatory slide. We're looking at the number of ticks over the 100 yards on the shady side versus the sunny side. These are the median number of ticks, and in the shady areas it was 6, and in the sunny side it was 0. The same results were obtained for each individual site separately, and for most cases for each individual week. So therefore these data we feel are very reliable, and therefore we can make the following conclusions. Ticks are not just in the woods at some sort of edge habitat. Ticks can be abundant at recreational sites, and they're definitely more abundant at the shady segment, the shady section of recreational sites. 95% of the ticks found were in fact collected were found on the shady transects. And therefore, it might be a good idea to include, and we will include that in our educational efforts about personal protection, that people who sit in shady segments of recreational sites protect themselves against tick bites by the usual techniques. This actually brings me to the presentation of Dr. Sam Telford, who is a professor and Lyme disease expert and has been for the last 25 years. All the techniques we use and learned are from him. He is a professor of Lyme disease and ecology at Tufts University and Harvard Medical School. And he will speak to us about ecology and management of ticks that spread Lyme disease. Go ahead, Sam. Oh, yes, indeed. We do want to finish. I am really sorry. We do want to finish our local research effort. In fact, it is my pleasure to introduce Mary Ablé, who will share the results of our latest Lyme disease survey that was conducted here in Dover during January and beginning of February. I am really sorry, Mary. I'm so looking forward to hearing Sam Telford that I couldn't wait.
Thank you. There have been 2 surveys in the past 4 years administered by the Dover Board of Health Lyme Disease Committee, one in 2009 and one this year in January. The slide above provides the details regarding the number of surveys sent out and the number returned. As you can see, participation rates were 31% and 22%. Now, naturally, the higher the participation rate, the better, but the rates in this range do definitely provide a basis upon which statistically competent conclusions can be made. And I'd like to say thank you to all who took the time and effort to complete and return the surveys. We have a one-sheet summary of the results available on the information table in the back for you to take home if you would like. One key piece of information we wanted to achieve from the surveys was a measure of the incidence of Lyme disease in the past 12 months. We asked this question in both surveys, so now we can make a comparison. In 2009, 26% of the households responding to the survey answered yes to this question. In 2013, that number was 11%. Now, 11% is still very, very high relative to the rest of the country. However, the data supports the conclusion that the rate has decreased in Dover. performing daily tick checks, wearing your protective clothing, using a dual repellent strategy, and staying away from high-density tick areas are all precautions you've been taking, and they've been paying off. So we believe that the likely factors for this decrease is education and awareness. This result shows that Lyme disease is a preventable disease, and our goal going forward then will be to continue to drive the incidence of Lyme disease towards zero in Dover. Another question we asked in the 2013 survey was in regards to damage and loss caused by deer. 26% of those participating in the survey provided a dollar figure for damage related to property, property or automobiles over the past 5 years. So that was about 127 of the 489 surveys we got back. So for fun, we added all these figures together and found that an aggregate total of over $250,000 was spent for this purpose. We also got information from the Dover Police Department regarding the number of deer automobile collisions. 38 were reported. Now, we don't know what percentage of deer automobile collisions are actually reported, so the real number is most certainly much higher. Okay. We also asked Dover residents to rank their concern regarding the ecological impact of deer in Dover. A ranking of 1 was for lowest concern and 5 for highest. When we tallied up the data, we found that 50% of Dover residents rated their concern a 4 or a 5. Taking it one step further, 75% rated their concern a 3, 4, or 5. Okay. We also asked Dover residents about the town-approved bow hunting program. In 2009, we asked whether residents were in favor of a controlled bow hunt on town land, and 88% of respondents said they were. This year, we asked whether residents would be willing to allow a town-approved bow hunter on their own private property, and 49% responded yes. Besides this 49%, an additional 5% wrote a special note on their survey saying that although they felt they didn't have enough private land to participate in the program, they supported it nonetheless. So we conclude that support for the town-approved bow hunting program continues to be strong. And with that, I'll turn over the podium to Jim Palmer, who will provide more information about deer management in Dover.
Good evening. My name is Jim Palmer. I'm one of 2 deer management agents for the town of Dover. Myself and George Junta, who couldn't be here with us tonight. Together we have helped develop and implement the deer management plan along with the Lyme Disease Committee, of which we are both board members and have been for over 4 years. Tonight I'm going to give a brief overview and update on the program. At the end, I'm going to ask for help from residents in support of the program. Hunting in Dover has been going on for generations. I've met families here in town where the sons are now hunting where their grandfathers once hunted. Methods on private property include both gun and bow hunting. The town-sponsored and managed program is a bow-only program. We have successfully completed our 3rd season, and as a result of the town program, we now know who the majority of our hunters are in our woods, and we have reduced illegal activity and trespassing. There are now rules and regulations in place for our town-approved hunters to follow, which further protect Dover residents. Here are some of the safety guidelines that we've put in place that we're very proud of. First and foremost, all hunters must abide by strict mass wildlife rules and regulations, whether they're on private property or they're town-approved hunters. We have additional town rules in place for the town hunters to protect Dover residents, keeping hunters at a safe distance from some of the commonly used trails. Our hunters have all been selected based on years of bow hunting experience. We accepted a lot of applications, and we primarily looked for candidates that had 20 to 30 plus years of bow hunting experience, and we selected those folks for the program. The Lyme Disease Committee and the DMAs then conducted a personal interview with all these hunters that we selected, and we even took it one step further. We asked the Chief of Police to provide some intel on these hunters, and he conducted extensive background checks, checking them for driving records, criminal history, as well as to see if they had any mass wildlife fishing or hunting violations in their file. Then TTOR required additional interviews as well for hunters on their land, and they also required proficiency testing, which all of our hunters passed. Our hunters have participated in the community. We encourage our hunters as they enter the woods to go on their hunts or on their scouting missions to get involved in property and trail cleanup. We ask them to leave the woods cleaner than when they found them, to take any litter or garbage that they find in the woods or in the parking lots out with them. They've also helped with ecology initiatives, including taking the day off from work to get involved in deer fencing issues, deer fencing projects on TTOR land. If you'd like more information on those, you can contact Mike Francis over at Powysset Farm of the trustees of the reservation. I wanted you to know that all our deer that are harvested are consumed by our hunters and their families. Nothing goes to waste. The DMAs, myself and George, have also participated in several regional town meetings aimed at increasing Lyme awareness and helping towns develop and implement management plans in towns like Weston and Medfield, who now have town hunts. We also managed the DPH intern tick density study and helped train the intern on safe tick collection, as well as provided the drag sites for her study. Lastly, this is where we're going to ask for your help. The town-approved hunting land is only a small fraction of the land in Dover where the deer live. I'm going to show you a slide of this in a minute that will be a good visual for you on that. As Mary said in her report, 49% of the survey responders were in favor of town-approved hunters hunting on their land. However, at the time we collected the surveys, we did not collect names and addresses of those folks, so we were asking the folks to come forward and contact the Board of Health. Here is that slide I talked about, the shaded areas in greens and purple. Those are right now the town-approved hunting lands. And as you can see, by all the white space in between, there's a lot of land where these deer can go and hide. And you've probably seen them out your kitchen windows and in your driveways. And we really need your help to open some of this land up. So in conclusion, going forward, we want to improve upon our results by expanding the hunting land to private property here in town. If you're open to hunting, and we can verify that your property meets mass wildlife and Dover rules and regulations. Keep in mind, not all properties will meet these rules and regulations, but we are asking you to contact the Board of Health to explore the opportunity to be more successful. Thank you for your time. I will be available in the back of the room for questions. Please enjoy the evening.
Thank you very much, both Mary and Jim. Our next speaker, now I finally get my chance to introduce Dr. Telford. As I said, he's a professor and Lyme disease expert with over 25 years of research experience at Tufts University and Harvard Medical School. And he's going to speak to us about ecology and management of ticks that spread Lyme and other tick-borne diseases.
Good evening. I'm here actually preaching to the converted, I hope, but nonetheless, it'll be useful to remind people, and maybe we'll get a few more converts. What I'm doing now is passing around some ticks. I go tick collecting every chance I get, and it was a nice day today, so I came an hour or two early and went down to Rocky Woods. And so the orange cap tube that you see has ticks that I collected in about 40 minutes. Separated by males and females, so you can take a look at what those look like. And the plastic bag with the violet ticks is what I collected over on Powysset Street or Road. There are 3 sites that I've been sampling there for a couple of years just to see what the tick numbers are like in roughly half an hour's worth of work. And so it is tick season again. And if we could, let's see. There we go. No, wrong one. All right. And I think Dr. Sam Donta will spend some more time on the actual clinical aspects of tick-borne infections. But just to remind people that even though Lyme disease is the most common infection and it alone is sufficient reason for us to want to do something and to do it pretty badly, people should be fed up by now. There are other infections out there, and in fact the deer tick itself transmits 5, including Lyme disease. And dog ticks, lone star ticks, woodchuck ticks, and there are even things that I call agents in search of emerging diseases that we find in ticks, but for which case reports do not exist. Therefore, there's no proof that they're actual human infections. All of these things can present very, very similarly. In the case of Lyme disease or Rocky Mountain spotted fever, you have a fairly characteristic rash. But when that doesn't appear, and the frequency of that is debated, these things appear as fever, chills, muscle aches, fatigue, and you can go from there. In terms, if you're a physician, you know how difficult it can be to actually pin down what is the cause of what we call fever, summer fever, or fever of unknown origin. And so, like I said, Lyme disease is sufficient for you to care, sufficient for you to want to do something. But it's not the only thing there, and in fact, 2 of those, deer tick virus and babesiosis, can be fatal. Next slide, please. We need to understand the life cycle of the deer tick if we want to do something environmentally, that is, to clean up around our environments and protect ourselves from risk of infection. So right now we're in adult tick season, and that's what you see in the vials that are circulating, and I do need those back. I'm going to experiment on them. They're around. The ones that I collected today are the same ones that emerged, sort of like cicadas emerging out of the ground, in October and November of last year. And they've been looking all winter for an animal to feed on, and that animal has to be large. It has to be dog-sized or bigger. So dogs, coyotes, foxes, bear, deer, even people. And if they're successful, they will swell up to nearly 200 times their size, drop off, and hide somewhere. We don't know where. It'd be awfully interesting to find out. They're not mechanically built in order to dig holes and go anywhere. They have to be somewhere under the leaf litter, protected from the elements. If they're not successful, they will look for animals, look for warm bodies all throughout the winter. If it's too cold for you to be out without gloves for more than about 15 minutes, it's too cold for the ticks. They're cold-blooded. They don't, they just fold up and hide when it's too cold for them. And so with all the snow that we had this last winter, it seems to me that they probably accumulated over the winter, and now they're pretty hungry and they're out looking. Typically, there are fewer ticks in the spring than there are the fall. I don't see any difference in terms of numbers between what I saw last fall and what I see today. Normally, I should see fewer. But a lot of people First time out in the sun in the springtime after a long winter, they'll brush up against some shrub, and they'll get 3 or 4 ticks on their pants, and immediately it's, oh my God, this is the worst tick year ever. It's not. These are the same ticks that are around in the fall. So if they're successful in feeding the fed female, the male doesn't take any blood, it's just there to mate with the female, the female will lay its eggs synchronously. It doesn't matter whether it fed in October or it feeds now. They will synchronously hatch, so the eggs are being laid right now. They will synchronously hatch in mid-July. And the baby ticks, the larvae, are literally the size of a period on the end of a sentence. And they are looking for things within a couple inches of the ground. Usually that's mice, shrews, chipmunks, squirrels, certain birds, and they'll even feed on deer and they'll feed on people. Now note, they're born uninfected. So at most, at that stage, they're a pest, like a mosquito. They're just something that causes an itch. But if they feed on an animal that has bacteria in it, the Lyme disease bacterium, they will pick up that infection during feeding. They can pick up that infection, despite what the literature says, from a lot of different animals. Usually it's the white-footed mouse, but it's chipmunks and shrews and even yellow throats and wrens and robins. If they feed successfully in August and September, they will drop off probably in a mouse nest or under the leaves somewhere and hide over the winter in an engorged state. And right about now they're developing as the temperatures get warm, and they'll molt to the nymphal stage or teenager stage of the tick, and they'll be out starting in late April, and most, depending on how cold it is during May, June is the worst month. They're really hungry during June. July, they'll start to taper off. And again, those times are approximate. If it's nice and dry during June, dryness kills ticks, hence the results of sunny versus shady. If it's really dry in June, the ticks won't last into July. But the nymphs are around in June, July. They will feed, again, on all of those animals, mainly within the first couple of inches of the ground. But if a deer beds down on top of them, they'll feel free to feed on the deer as well. They'll molt, they'll develop to the next stage over the summer, and then become the adults for October and November. So it's at least a 2-year life cycle. These things have to exist from larval to nymphal stage over 8 months without taking a blood meal. Next slide, please. And so it's really important for people to realize that there are 2 factors required in order for us to have contamination in our backyards, risk in our backyards. That is producing the ticks, and that's a function of where the female tick gets its blood meal. So if you have a lot of dogs around and they're not cared for and they're not front-lined or something like that, they might drop ticks in your yard. But in general, most of the blood meals come from deer. Each female that feeds successfully will lay 2,000 eggs. So, production of the tick and infection of the tick. And that's a whole lot of different animals, and you can tell right away what's going to be the most effective target for us, the most cost-effective target. It's going to be hitting tick production. It's not going to be going out and trying to kill mice or shrews or birds. And in fact, you can have the successful feeding of one female will compensate for the death of 2,000 immature ticks, larvae or nymphs. So again, more cost-effective to target production. It's the same with mosquito-transmitted infections. West Nile virus or Eastern equine encephalitis, you don't go after the birds that are the reservoir of those viruses. You go after the source of the mosquitoes. you clean up, you target the breeding sites. You only go after the adults that are infected by spraying only when there's an emergency. Next slide, please. And in fact, this is borne out by when the cases appear. So it's an old slide from the 80s, but if you plot out when the cases of babesiosis or Lyme disease appear in the hospitals, most of them come in during late June, early July, middle July, and some in August for either babesiosis or Lyme disease. And note that very few infections are seen from late August, September, October, November, and through the winter, which is kind of odd because, remember, the larvae's born uninfected, it picks up infection from a mouse or a bird, feeds and turns into the nymph. The nymph is infected at a rate of about 20%. So if you go out and collect ticks during June, 20% of them are infected. Those ticks have to feed in order to develop to an adult. They feed again on something that has the chance of infecting them. So they've had 2 chances to acquire infection, and the infection rate in adult ticks is generally double that in nymphs. So if a tick is crawling on you, you have twice the chance of it being infected now as in June, but we don't see any cases during that. Why is that? And the other graph simply shows that if you go out and trap animals and count the ticks on them at various stages of the year, you see that larvae appear generally around July and disappear in October, and the nymphs appear in April and disappear around July. Just as this is how we piece the life cycle together, and that's how we incriminate what is actually transmitting to people. We look at those, we superimpose the graphs and say, well, if you give Lyme disease roughly a week to 10 days to appear, that means most people acquired infection sometime in June. Next slide, please. So that's an important point to remember. June is the riskiest month. There's another reason for there's the main reason for us not seeing Lyme disease as frequently during the winter months is because of what we call the grace period. It's an adaptation to an extended life cycle. Remember, if a larva picks up infection from a mouse in August and September, the infection doesn't have a chance to get out till May or June. That's 8 months. If the bacteria is metabolically active during those 8 months, it would eat up the resources, the fat supplies of the tick, and cause it harm. It would be a negative fitness effect. So what the bacteria does is go dormant. It goes to sleep during those months. And when the nymphal tick comes out and starts looking, it's at ambient temperature. It gets on a mammal, skin temperature is 37 degrees centigrade. That's a big change in temperature from ambient to your skin. And it tells the bacterium, wake up, it's time to get out. The blood coming in helps it to replicate. And so it takes 24 to 48 hours for that to happen, for activation and replication to the point that they become infectious. So, and this is a general feature of tick-borne infections. Other than tick-borne encephalitis virus or deer tick virus, there doesn't seem to be a grace period. It's in the salivary glands and it's ready to transmit the moment the tick attaches. So there's a good reason for this so-called grace period. It's biological. There's actually changes in gene expression going on at the same time. Very nice molecular biology being done there. And the bacteria actually migrates from the gut of the tick to the salivary glands. So most people should know that a tick isn't like a mosquito. It's not going to feed instantaneously and then fly off. It's going to be on you for a period of days. For the nymph, it takes 4 days in order for it to create a pool of blood, soften its skin in order to take what we call the big sip in the final hours of the blood meal and swell itself up 20 to 200 times. The female tick takes 7 days. And it's midway through the blood meal that's most important for infectivity. So for nymphs, that's roughly the 2nd or 3rd day of feeding. For the adults, it's probably 3 to 5 days of feeding. The other thing is that's a nymph and a fingernail. An adult tick, as you can see, is much bigger. You'll see it and remove it before it's had a chance to spit enough bacteria into you. Finally, remember how you dress now versus how you dress in June. The ticks have a harder chance of getting up underneath your clothes right now. So all of those factors mean that adult ticks really aren't as much of a problem as nymphal ticks during June. Next slide. So a little bit of history helps us understand what we can do as communities to try to reduce the contamination around our homes. Why coastal New England, and why did these infections appear in the 1970s? Some sort of threshold was reached then. We chopped down the forest. The colonialist, each house required an acre of wood to heat the home during the wintertime. And so very quickly, much of New England looked like the top right picture, which is Nantucket Island in 1880. The bottom picture is Nantucket Island from the same vantage point in 1980. Now you have thick shrubs. Before, there weren't any white-footed mice, and there were no deer. Where are they going to hide? Now, you could walk right by on that road, and there'd be a deer bedded down, and you wouldn't even know it. And the ticks require it. The ticks need 85% or greater relative humidity in order to survive more than a week. It's now about 30% relative humidity in here. You take a vial of ticks, such as the ones that I'm passing around. You leave them on the tabletop for a week, half of them would be dead. So the brush is very important. We obviously are using the habitat more, and the deer have gotten much more dense than they ever were. Of those three, what can we manipulate? We can manipulate habitat if we really wanted to, but remember, we want to try to prevent as much tick reproduction as we can at the same time. Next slide, please. So this is my mantra now. We know how to kill the ticks, we just can't get people to do it. Next slide. This fellow is the chief of Lyme disease studies at CDC. Everyone should know this book. I know that the Board of Health and the Tick Committee here has sent around a link to this, or has prominently featured it. It's available online. It's free. You can download it. It has everything you need to know about how to protect yourself as well as reduce contamination around your homes. You should have a copy. You should read it, and you should take the hints from it. Next slide. What do we do at the level of individuals? What we do at the level of individuals is not the same thing as we might do at the level of communities over time. These things have to be done every day. Repellents and toxicants, use off. Coat your clothing with permethrin. Permethrin won't repel ticks, but it will, if a tick crawls across it, it will die in 3 hours. Appropriate clothing. I've managed to avoid Lyme disease simply by tucking my trouser cuffs into my socks. That helps, but you look like an idiot. And you don't want to do, most people don't want to wear long trousers during June. Tick check, extremely important. Get in the shower, feel yourself for new bumps. You're not going to see these things. You got to feel for new bumps. Even better, have someone check you in the shower. Education and awareness. If all of you pick up a few of our message points tonight, that may help you reduce your risk this coming summer. Awareness, if you have a fever and you don't have a runny nose and it's the summer, go see your physician. Habitat avoidance, if you have HIV, you're splenectomized due to a car accident or some other condition, or you're on chemotherapy for malignancy, don't go to, don't work in your yard during June. Or if you do, wear appropriate clothing and use repellents and toxicants. And then finally, reduce sources around your homes. Clean up the brush. The ticks need brush to hide. Put your bird feeder off in the corner. Don't promote deer around your yards. And so all of these things, though, have to be done every day. Next slide, please. But at the level of communities, hopefully what we can do is manage things so that over time, you don't have to be vigilant every day. You can manage your habitat. You can clear the brush. Burning is a wonderful thing if you can do it. We need to try to kill Smokey the Bear. Fire is actually a good thing for forests, and it clears up all the leaf litter where the ticks hide and thrive. Desiccants, if we mulch it, if we put down stuff that dries out the land, even better. Obviously, education and awareness. If you know where these things are coming from, you can target your resources effectively. Spraying is not a bad word. The new generation pesticides, the synthetic pyrethroids, are actually very effective and very short-lived and not toxic to mammals and most other animals. You can't apply them near wetlands, but certainly if the FDA will allow you to put permethrin in a shampoo on your kid's head for 30 minutes, it's a pretty harmless substance. Most people have this fear of spraying because they were indoctrinated by silent spraying. Silent spraying was 50 years ago. Your dad was driving a Studebaker. Today, we're driving much nicer cars. It's the same with insecticides. Things have changed. Host-targeted acaricides. You can take cotton balls, put synthetic pyrethroids on them, dry them out, put them in toilet paper tubes, and put one out every 15 feet or so perimeter of your yard. Mice will pick it up, take it back to the nest, it'll rub off on their fur, and they'll be cleaned of ticks. So what that does is reduce the amount of infected ticks in your yard. Deer targeted acaricides, a bushel of corn in a hopper. The deer, in order to get at the corn, have to pass these paint rollers, which have an oil-based permethrin. It coats their fur, and the ticks die on the deer. So it targets tick reproduction. These things work very well. It's just you got to put a bushel of corn in every 2 weeks. You got to take all the old corn out, and you got to tolerate all the extra skunks and raccoons and gulls and crows and rats. So you may be substituting another problem, not to mention the fact that they're really expensive. So there are all these things that can be done, but let's spend the final few minutes on deer reduction and the basis, the scientific basis for that. Next slide, please. Deer feed the most adult ticks. That is the same kind of tube, the orange tube the orange cap tube that's passing around, but it's got 99 engorged female ticks from one buck at the Crane Reservation in Ipswich in 2004. 99 times 2,000 is how much? So why aren't we up to here in ticks? That's something that I'm actually trying to study. But you can see that in these sorts of studies have been done elsewhere. If you look at all the potential hosts for adult ticks, 90% of all the adult ticks are feeding on deer. Next slide, please. I speak with some sense of experience because as a doctoral student, I did what was known as the Great Island Experiment. We reduced deer from 35 to 50 animals per square mile down to fewer than 8 on a peninsula off of Cape Cod. They came on and off as they pleased, but starting in 81 and 82, the deer were counted. Fish and Wildlife came in after exploring all sorts of other options, including moving them, getting the NIMBY attitude, not in my backyard, please, and decided it was far better to reduce them by shooting. Brought the deer numbers down, and they have remained down. The caretaker was asked to keep the deer numbers down to fewer than 8 on the property. I went out and trapped mice from 1985 and through 1994 and counted ticks on them, and you see one slope going way down on larvae and also a gradual slope for nymphs on this Great Island site in Nantucket. If anything, things were increasing. I continue to go out there twice a year to collect ticks, and the numbers remain low, even in the presence of coyotes, foxes, raccoons, skunks, and possums. So for some reason, they have not jumped onto other animals, and there is some relationship between, there's something that tells a deer tick that it's a deer. Not only that, we did very aggressive case detection. Right after, before deer reduction, we could easily see 1 to 3 cases a year. I would bleed people every July and August for 10 years and detected asymptomatic infections that way. And so we know for a fact that deer reduction led to diminished incidence of Lyme disease in this site. 80% reduction in risk is what we calculated. We didn't eradicate the deer. The deer ticks are not gone, but they're greatly diminished, back to what they used to be. Next slide, please. In the interest of time, let's move on from the slide. There are a number of studies that replicate these results. That is, if you reduce deer, you see reductions in tick density. This one is illuminated because a lot of people try to use this as an argument against deer reduction. Oh, they reduced deer in New Jersey and saw no effect on the ticks nor on Lyme disease incidence. Fact of the matter is they reduced it from 45 deer per square kilometer in 2002 to 24 deer per square kilometer in 2005. The conversion factor is 2.6. So you're going from 120 animals per square mile down to about 60 animals per square mile. You need to get it down to below 10. So they did nothing really for deer reduction. And I actually argue that their table 2 does show effects on the ticks. And in fact, the 2 dotted lines that you see on the graph, the top one is the untreated site and the bottom one is the treated site. And I think the difference between 1 nymph and 4 nymphs is actually a lot of difference. Next slide, please. So the data is consistent with deer serving a causal role in the density of ticks. This is what happened. This is what's happening around us. 15 years ago, I wouldn't be here. I'd laugh if somebody asked me to speak here 15 years ago, because there were no ticks here to speak of. Over the course of 6 years there, this is Mass Department of Public Health data that we analyze of case reports, towns with more than 5 cases reported each year, and then extrapolated for areas around them. You see the amount of red has increased dramatically in the span of 3 years. Juxtaposed with a map of mass wildlife's setbacks. That is, there are communities where hunting is very difficult because you need to be 500 feet away from an inhabited house unless you have written permission. So you cannot hunt archery or shotgun within 500 feet of an inhabited house. And you'll notice it's awfully hard to find space to hunt in eastern Massachusetts. I don't think it's coincidence that you see the red that you do there as a direct result of not being able to manage deer. Next slide, please. And in fact, Dover, to its credit, and Medfield as well, has been the leading edge of communities being fed up trying to do something by initiating and running a deer reduction program, trying to open up lands and trying to actually bring things back to where your grandfather would remember them. That is, they could walk in the woods for miles and never see a tick. That's what we would like to do, is bring things back so our children can experience that. It's too late for us, but maybe we can do something for our children. Finally, deer serve as blood meal sources for a lot of things. 90% of the blood meals for mammal-biting mosquitoes come from deer. And that's done, these studies actually measure the DNA or protein left after a mosquito lays its eggs and can determine precisely what animal, down to the species, that a mosquito fed on in its previous blood meal. 90% of all human-biting mosquitoes, the ones that currently are starting to, in the next 2 weeks, you're going to see what we call snowmelt mosquitoes. Most of their blood meals, the eggs from which they're hatching, were laid last fall as a result of blood meals from deer. Next slide, please. But remember, it took from colonial times until 1970 for the problem to develop. Nothing we do is going to bring it back within the span of a few years. Nor are we ever going to clean the environment. What we can hope is the blue dots signify risky areas because of contaminated yards. What we want to try to do is bring it back to the right where those risky areas are smaller and smaller. And that's going to take deer management, habitat management, education, and a commitment by communities, such as what the Dover Board of Health and the Tick Committee has done here. So with that, I'll pass the baton to Dr. Donta.
I'd just like to remind you, if you have any questions for Dr. Telford, please write them down on the green cards. And after Dr. Donta has had a chance to speak, then question for him on the yellow cards, please. And with that, I'm delighted to introduce Dr. Sam Donta, an infectious disease specialist over many decades who is working at in Falmouth, and he will speak to us about Lyme and other tick-borne diseases, symptoms, and treatment options. Now we've got to go back to.
Yeah, I think he's going to do it.
Yeah. Maybe you can even.
There we go.
There it is.
Good.
Oh wait, you can't. There you go. Now it should work.
Well, let me see if it does. There we go.
Perfect.
Thank you, Barbara, and the other organizers to invite me to talk about some aspects of Lyme disease and other tick-borne diseases. I will say a few words about some of the other tick-borne diseases, but the focus will be on Lyme disease as the major ongoing problem. There's been an evolution in our understanding of Lyme disease. As with any other disease, when you first discover it, you find the typical parts, and they were described in stages, oversimplification a little bit, and what was described as Lyme disease at the beginning still is that. That is, you can have a big rash following the bite of a deer tick that is infected. You can develop multiple rashes within the next 2 or 3 weeks, and then a number of weeks or months later develop a Bell's palsy or cardiac block or a big swollen joint. And when that was first discovered, that is, a group of children from the East Haddam Old Lyme area, it was an arthritic presentation. And they went to the rheumatology department at Yale, and it was investigated. It was thought to be a form of juvenile rheumatoid arthritis to begin with, but it didn't quite fit. Eventually, though, it was discovered that ticks preceded this, and through the work of one of the infectious disease fellows, Ellen Barber, and the senior person at the Rocky Mountain National Labs, Billy Bergdorfer, he discovered the bacteria, the spirochetes, from within the midgut of the ticks, and the disease was named, the bacterium was named after Bergdorfer. There are other genotypes of Borrelia. They fall into a very interesting group of organisms. In this country, we really didn't know any Borrelia. In South America, there's relapsing fever. So we actually have a precedent for an illness that has staying power. And I think this is an important part of this, because the disease understanding became a problem, and polarization developed, because we still to this day do not have enough information upon which to make the types of dogmatic statements that are being made, let's say, on both sides. I try to be a bridge, if you will, trying to understand and use science best I can. So what I'm going to do is just go over a few aspects here, some of the clinical aspects, some of the laboratory studies, and some of the treatment. Yeah, right. There we go. And the stages were described, and they're still described as now early disseminated, and late or chronic Lyme disease. And I'm going to end up focusing on the late and chronic, because that's the biggest area of misunderstanding or lack of understanding. It's easy to understand early Lyme disease if you've already had a rash. It's important to understand, though, and the SmithKline-Beecham vaccine study in the 90s showed us that half of the rashes are not typical bullseye. And that was from culture data. So there are still some physicians who think that you have to have a bullseye rash in order to do that. in order to have Lyme disease. And the percentage of rashes that occur, it was thought initially was 85%. I don't know, it may be half or less. And these rashes are generally asymptomatic. You may not see them if they're under your armpit or if they're on your back. They may be itchy, they may not. They may present as a bruise. I don't have pictures here to show you, but this creates a big difficulty in trying to diagnose it when you do have a rash. What do you do when you have a tick bite itself? Dr. Telford described about that the ticks generally need a certain amount of time to get their mechanisms together. When you find a tick, if it was just on you, that's easy enough. You remove it, even if it's attached, and it's very unlikely that it'll be able to have transmitted the infection by then. Nonetheless, there are confusions, controversies as to what to do about a tick bite. If you do have the tick and you don't know how long it's been on you and it is attached and you remove it, you can have it tested. University of Massachusetts in Amherst has a lab that tests it 3 times a week for $35 or $40. Does that mean you don't have another tick that you didn't see? But you're concerned with the tick that you found. And so that's one approach. But many people will just throw the tick away. Let's flush it down the toilet. So now what do you do? An attempt to address that was made by infectious disease people from the University of New York in upstate. And they suggested that maybe if you take 2 doses of doxycycline, that helps. That's not surefire, but that may pacify both the doctor and the patient. I think the important thing is that you need to see then in the subsequent week or 2 or 3 if you start developing symptoms, whether you've taken the doxycycline or not. But that's one approach you could do. There are some doctors who prefer to say, wait until you have symptoms. That's a little bit more tricky because you may not develop symptoms for weeks or months or years. And so how do you know? Our existing blood tests are not that sensitive. And if you were to do blood tests, you probably have to wait a couple of weeks to see if your immune system, your circulating antibodies, have begun to make certain antibodies that would tell you. But the bottom line here is that you need to rely on the symptoms. And at that point, it gets difficult because the Lyme screening tests, these are indirect tests. They depend on asking the question, has your circulating immune system mounted a response against the bacteria? So are you able to find it with the screening test? Will the more detailed Western blot give you more answers? And like circumstantial evidence, when you have it, it's helpful. But does a negative test mean you don't have Lyme? And the answer is no. But how do you then judge what to do? And these are individual decisions that need to be made by physicians with their patients. And I've talked a little bit about this, and in the interest of time, I'm going to move ahead. But if you start having symptoms, then it appears that especially early on, in the first month or 2, if you develop a flu-like illness and it's summertime, think about summer flu, is not usually influenza, and flu is a misused term. Influenza is a respiratory condition, but people refer to diarrhea as flu. But this is a illness that you feel like you're achy all over. You don't necessarily have a cough, but you have a fever, and you're tired, and it's the wrong time of year. So you need to think, can it be Lyme? And you'll be fortunate if the blood tests are positive, but many physicians will go ahead, and I think appropriately, treat with 1 of 3 different antibiotics. At that point, amoxicillin, a penicillin derivative, or doxycycline, or if you're allergic to penicillin, a cephalosporin, as it's called, is generally effective. How long to treat? Arguments of 10 days to 2 weeks to 3 weeks to 4 weeks. I use a rather common sense approach, treat until your symptoms are gone. So for anybody who says, I've treated you for 3 weeks, you no longer have Lyme disease, that statement is not substantiatable. And I think that's the problem that we're having now, and that leads me into the next phase to talk about the persisting or chronic disease. I'm going to skip back to this. This was the early description that was called late Lyme disease, the Bell's palsy, meningitis, carditis, or the arthritis. And that, by the way, became an easy way to diagnose it. And the interesting thing about that is that short-term treatments generally work. And when you look at these patients and compare them to the group of patients whose immune response is not as robust, the thinking I have is that maybe the patients that did mount a good immune response limited the amount of bacteria that could be established and generally have a more favorable outcome. And that is the case when you study patients with this classic form of Lyme disease. Now, the difficulty is, how do you know if you don't have a full array of immune responses that that patient has Lyme disease? Because we don't have a test to tell us the infection's still there or not. We know that if the initial rash was there, you could theoretically culture, not from a routine lab, but in a special lab. You could find the bacteria in the rash, and you could find it in the bloodstream on and off for a number of weeks. After that, you can no longer find it. Neurologists still are under the impression that you do a spinal tap, and if it's negative, it doesn't have this ratio, you don't have Lyme disease. That is not accurate. Like circumstantial evidence, again, when you have it, it's helpful. A negative result just makes it more difficult. So where are these bacteria? Where do they go? We know they're under the skin in a number of animals, and more recent studies in monkeys show that monkeys who were experimentally infected, they weren't infected with deer ticks to begin with, they were inoculated with the infection. Nonetheless, they developed what was thought to be Lyme disease and were treated. One group with IV antibiotics and doxycycline, another one with doxycycline. And 8 months later, they couldn't find the bacteria, but they put sterile, uninfected ticks on the monkeys and were able, after letting them feed, transfer. They found the bacteria to guinea pigs and hamsters. So they are persisting, but they're persisting, and that study hasn't been done in humans. I think there's a study at Tufts that's supposed to be looking at that. And so, where do you get the ticks from? But are they sterile? Are they sterile?
Well, they're not sterile. They're specific pathogens.
Yeah, well, that's what you have to do when you're doing an experiment, because otherwise an institutional review board will say, wait a minute, you're taking ticks and you're putting it into people, you're going to transmit who knows what. So that's an important study to do. My own thinking about what happens is that that is not enough to explain any possible persistence. With any other infection that I know of, especially bacterial, fungal, parasitic, you have to find a reservoir, a protected place. And that's usually inside cells, intracellular. And the data that we've amassed with certain treatment trials speaks to that likelihood. But I'll go ahead and get into that. The problem with chronic Lyme disease is, what do you mean by chronic? Well, you mean it's ongoing. So let's not play with the term and say that's a derogatory term. Because some people say, well, you're chronic. But chronic fatigue is a legitimate description of an illness that we still don't have a unified or even a group of pathogenic agents, presuming that it is an infection. Nonetheless, this group of illnesses all have the same combination of symptoms. The fatigue, the arthralgias, the cognitive, and then a group of what I call minor, which may not be so minor for a number of patients. Headaches, eye symptoms, palpitations, paresthesias, tremors, jaw pain, tooth pain that's unexplained. And when you look at chronic fatigue syndrome, you see that the official definition, going through a few changes, is that you have to have unexplained fatigue for more than 6 months, and if you have to have 4 out of 8. and look at some of the symptoms there, and look at what fibromyalgia has. It's the same thing. When we studied Gulf War illness, it was based, this was the first Gulf War, it was based on the chronic fatigue syndrome, and it's unlikely that those patients had Lyme disease, but we don't know what they had. They're still wondering whether it was chemical exposure. How can that do it? Is the chemical damage something? Is it persistent? And when you look at Lyme disease, chronic fatigue syndrome, fibromyalgia, Gulf War illness, and you look at the symptoms, you can't tell them apart. Now here's a question. Does that mean that anybody who is diagnosed as chronic fatigue syndrome has Lyme disease? Does that mean that anybody who has fibromyalgia has Lyme disease? I think some of them probably do, but I don't currently have a good idea of percentage-wise. Are we talking about 10%, 50%? My thinking is that if you have a complex of symptoms that include these, and you live especially in an endemic area, the possibility of Lyme is sufficient that you need to pursue it. And how do you pursue it? You pursue it with a straightforward analysis. The physician does a good history, exposure history, travel history. They do a physical exam. They do a variety of routine blood tests and specialized blood tests. And it isn't a diagnosis, shouldn't be a diagnosis of exclusion, but in part it is. You do the rheumatoid test, well, it isn't rheumatoid arthritis. It isn't lupus. It isn't this or that. So when you have blood tests, though, that suggest that you have been exposed to Lyme, my approach has been to give a therapeutic trial to see if the patient's symptoms begin to respond. And I tell the patients, if you don't have Lyme disease, nothing's going to happen over the next 2 or 3 months. And let me get into that just a little bit further. So what are the cause of the symptoms of persisting symptomatology? It seems to me there are only a few possibilities. Persisting infection. Are there some kind of autoimmunity responses? Most of the field of rheumatology is based on that your body has reacted to some unknown substance that cross-reacts with certain cartilages or other connective tissue diseases. And there is evidence that that takes place. My question in that regard is, what was the trigger for it? What was the initial? Was it an infection? Was it not an infection? Was it a chemical? And there is some thinking that with autoimmunity, that maybe you've killed the bacteria and there are residual debris. And there is, on the outside of the bacteria, the Lyme bacteria, the Borrelia, they do have complex combinations of lipids and proteins and sugars. And that is a possibility that your body then reacts to that. And then you might have neural tissue, myelin, other things that have those same components, and that you are reacting in a cross-reacting fashion to that. And that may be part of the answer. My thinking that even if there is some autoimmunity, if you get rid of the underlying driver, the source of it, that will go away. And that's my observations, that that seems to happen. But I'll come back to that. Now, post-infectious sequelae, what do I mean by that? Well, is there damage? Or is there something unknown that we haven't thought about? I don't think it's damage. It's not like having a stroke where you just have lost the function of a limb. These symptoms fluctuate. They don't always have to have a pattern, but some are month to month or week to week or season to season. And there are ups and downs. It doesn't sound to me like it's a result of damage. And because the symptoms and some of the signs can be reversed, that I think it's unlikely to be damage. So I think we're left with is there a possible infection? Now, this leads me to discuss briefly, where do the bacteria go? They probably do go under the skin, and there's experimental evidence in animals that there are residual bacteria, maybe not alive, but maybe some of them are alive. And if they go into intracellular compartments, how do they cause their illness? Well, when you look at generally bacteria in general, let alone other organisms, there are really only two possible mechanisms. It's not their mere physical presence. These are microbes. So I don't think just having a bacterium in a particular compartment or under your skin is going to cause you a localized symptom. So they either contain substances or produce substances, and they may do both. And some of the symptoms to me do resemble toxic kinds of symptoms. If you think about how many diseases, infectious diseases, involve toxins, you have diphtheria, you have tetanus, even with the streptococci, the staphylococci, there are toxins that are elaborated that are part of the pathogenesis of the disease. But there are also components of the bacteria themselves, endotoxins, that may also elicit a body's response to that product, and you get symptoms. So I'm prepared to believe, until we actually know, that there may be a combination of events going on. And then I did talk about the autoimmunity. Now the blood tests. I talked a little bit about it. These are indirect tests, and you can't, I mean, if I want to ask you if you had German measles and you don't know, we can ask your mother, but otherwise we can do a blood test, and it might show that you've been exposed. Well, does that mean that you're sick with German measles now? No. So these blood tests are really not reliable indicators of activity. To that end, we look at general two types of circulating immune responses. The Ig stands for immunoglobulin, which is the chemical term for antibody. The G antibodies are the long-term memory antibodies that we keep, and hopefully they provide us with some immunity. The M antibodies are the first ones to come up, and they should go away when you're done with the illness. And not just my observations, but others as well, show that many patients with chronic relapsing symptoms continue to have IgM reactions. Now, the recommendations for interpretation of the blood is also a problem. The field has tried to, in general, simplify things so that doctors and patients just aren't overly confused. But in the attempt to simplify things, I think they're oversimplified. So that nobody wants a doctor to say anybody who has chronic fatigue might have Lyme disease. It gets tricky. On the other hand, you can't be making statements you don't have Lyme disease because you don't have a big swollen joint, or your blood tests, you didn't have 5 out of 10 bands, or you didn't have 2 out of 3 bands. And then some of the logic is mind-boggling. For example, the recommendations is that IgM is positive if you have 2 out of 3 bands, but if it's already ongoing, it's a false positive. What? Where did that come from? How is it a false positive? Who made that up? There are people who make up stories on both sides, if I might say. So that's what I was talking about. The Western blot is a more detailed way to look into it. The MRI can show, but not in many patients. In our studies, about 15% of people have these spots that can be similar to spots that MS patients have. Now, MS is really a different disease, but there are overlaps in symptoms which make it difficult, and it's part of the differential diagnosis that a doctor goes through in trying to understand it. The brain SPECT scan, though, this is like doing a bone scan, but it's on your brain, shows some abnormalities in 75% of patients. And what's nice about this in our published report is you can reverse those. So it is, let's say, an objective sign. Physicians want objective signs. They don't want to hear you with your symptoms and you look good, but you're telling them, I'm aching, I've got fatigue, I've got these crawling sensations on me, my brain is not working right, and what's the doctor do? So he goes by the guidelines that he's learned, and I think they're oversimplified. And I think one of the tasks of our mass commission is to try and educate doctors to become a little more educated on some of the complexities of the disease. We don't want them to automatically assume that everybody's got Lyme disease and they have to be on treatments forever. But we need them to understand that we need more research. We need an active trail marker if we can find one. And these are just some of the data from there. Now, what about antibiotic treatment then? We know it works in early disease, and 2 or 3 or more anecdotal studies said, well, I treated the patient with doxycycline for 30 days, and I treated them again for 30 days, and they didn't respond. So it's inappropriate, though, to say that they don't have an infection anymore. You just don't know at that point. You might think that that might be a possibility, but you don't know that. So I learned very early on when I was at the University of Connecticut and I saw my first Lyme patient. He had been treated with doxycycline a couple of times. He had these flagrantly positive Lyme tests, so there was really no question he had been exposed. The question was whether his ongoing symptoms were due to Lyme. And he had had doxycycline treatments. Well, I'm old enough that I know when tetracycline was first introduced, before doxycycline, that its pharmacologic properties were different than doxycycline. So I said, doxycycline might be a little bit what's called protein bound, whereas tetracycline isn't as protein bound. It's a higher dose. I treated him, and subsequently, in a number of studies that are published too, tetracycline works much better because of pharmacologic properties. So one has to be careful then about drawing conclusions about something not working, therefore you don't have it. And so we need to understand better what antibiotics work and what don't. In the test tube, there are many antibiotics that are active against the Lyme bacteria. But the question is, can they get to the site? We know this from a variety of other infections, that you can't use an antibiotic that doesn't get to a particular site if you hope to eradicate it. And I think this is the same issue. And what I learned from this, because I was perplexed why the erythromycin group of antibiotics weren't working in people when I know they get into all the cellular compartments and they were very active in the test tube. And it took me a while to figure out why that might be. I'm going to come back to this in the discussion period. Let me just come, I'll hold that thought. And let me come. Ceftriaxone, the IV drug that many patients continue to think will cure them. And that perplexed me for a while, and I've been there, done it, if you will. And I've treated a number of patients with ceftriaxone only to see them go back to square one. And yet I know ceftriaxone doesn't get inside cells, both experimentally and in people, inside cells. So how is it that people do feel better while they're on it? Well, a few years ago, as part of Lou Gehrig's disease research, they found, when they were doing what's called a molecular library search, looking for compounds that the presumptive defect in Lou Gehrig's disease was a absence or a downregulation of what's called a glutamate receptor. There's the neurotransmitters, there's a variety of them. One of them is glutamate. That a compound that allowed you to upregulate the glutamate receptor, ceftriaxone, was at the head of the list. So a light bulb went off in my head. Maybe you're allowing symptoms to improve, like taking aspirin for a fever or an ache, but without getting rid of the infection. Because we know ceftriaxone works, if not only, best on multiplying organisms. And there's no evidence that these organisms are multiplying in the body. I mean, you can hear lots of stuff on the internet that people, it does this, it does that. Those are speculations, and they don't have the substance that we need to have. So I think that probably explains why ceftriaxone gives you transient relief, but you keep getting symptoms and you don't get rid of them. What I think is that you get rid of them with either the tetracycline, used over a long enough period of time, or these antibiotics called macrolides. Now this is just a cartoon from somebody else that depicts how organisms get into cells. They either induce the cell to take it in, and what a clever thing for the Borrelia to use a nerve cell, because the body isn't going to get rid of a nerve cell. And it can stay there forever from that point of view. But anyhow, it goes through different compartments. And different organisms, we know from other organisms, mycoplasmas, mycobacteria, legionnaires, they'll settle into a compartment and remodel it to their liking, change the pH of it. And that gave me a clue as to what's probably going on. And I'm not going to go through this with you, but research from Dr. Raoult's lab in Marseille, when I saw that you could change the acidity inside the cellular compartment using specific either chloroquine or a chloroquine derivative. You can't give enough ammonium chloride to somebody, or amantadine. And would that then allow the macrolide antibiotic, the erythromycin, to work? And the first patient I did that on had been on Plaquenil. This is hydroxychloroquine, which is related to the chloroquine, which does change the pH intracellularly. She was on it for 2 years for seronegative rheumatoid arthritis. I did a Lyme Western blot. She had IgM reactivity against several proteins and IgG reactivity. I added clarithromycin, which is Biaxin, to that, and within 2 weeks she started to improve. And finally, by using that, and I think that allowed me, and I don't want to be definitive about this, but my model here is that they probably do occupy an acidic compartment inside cells, likely nerve cells. And that's why the data combining the hydroxychloroquine with clarithromycin show good results. Now, this is not instantaneous. And I learned over the years, I've been doing this for 25, 30 years, and having a science background, I hope I begin to ask the right questions. So my model right now, and my understanding is that it may take 2 or 3 months to begin to see a good outcome. Now you can argue, well, maybe they'll get better by then. Yeah, when you look at people who haven't been better for months and months and months, and coincidentally with medicine 2 to 3 months into it are starting to improve, there's a little bit of a probable cause and effect. Nonetheless, I have not proven this. And I would love to do an NIH treatment trial, but then that's another whole story about what treatment trials were funded and which ones were not. And the answer is they don't fund any more treatment trials. Depending on how long you've had it, the less time you've had it, and this is kind of common sense, the more likely you are to be cured and improve. Then the longer you have it, the less likely you are to get a complete cure. So these are observations. I'd have to admit these are not the results of controlled treatment trials, but nonetheless, I'm comfortable about it. I would love to do a controlled treatment trial to prove the point, because that's going to be the way that you convince physicians of the science of it. So right now, I'm an artist, if you will, from that point of view. There are a variety of other suggestions, hyperbaric oxygen. We go through, some people are getting intravenous peroxide. They're using microwave machines. They're using a variety of supplements. And here's another little tidbit. I didn't have any problem with this until I observed that people who were on a lot of these supplements weren't getting better as fast or as complete as those who weren't. And so I said, well, maybe the extra vitamin C, which is a strong acidifying agent, was keeping that compartment acid and counteracting the effect of the hydroxychloroquine. So I asked people humor me about not taking extra vitamin C. And the B vitamins are another story. How can B vitamins be bad? Well, it turns out, I think your body is trying to, with any infection, especially a chronic infection, starve it to death, if you will. It withholds iron. Well, it's another story with these bacteria. They're one of only 2 bacteria that don't use iron. It uses manganese. This is one clever bacterium. But anyhow, the body's trying to starve it to death. Some people then have a mild anemia, and they're thought, oh, well, they're iron deficient. No, they're not iron deficient. It's the anemia of chronic disease kind of a thing. Well, the genome for the Borrelia burgdorferi don't contain sequences to synthesize B vitamins. So they're the only bacteria I know of that don't. So I'm thinking maybe they're trying to get their B vitamins, their other materials from the body. So I tell them, humor me about not taking B supplementation. And that's not something I can easily study. I don't ask them to change their diets. These are kind of suggestions based on my expression. So I think we'll end now with the questions about We still don't know where the bacteria go. We don't know if they're still there or not. We need new tests to document that the infection is or is not there. Vaccines are the most important way, aside from preventing the tick bite. And there have been attempts. Actually, the SmithKline-Beecham vaccine was successful, but it ran into some issues with some patients with Lyme, knowingly or not knowingly. They had some arthritic complaints, so they got worried and they withdrew it from the market. Now, there is still some work, and more work should be done on various components of the Lyme bacteria and whether there's a toxin or not. As far as the ticks, the main thing is to remember the Babesia is a malaria-like disease that destroys your red blood cells and your platelets. And when a person comes in, especially in the summertime, with a high fever, with sweats, they're fatigued, they did or did not know about a tick bite. The lab should be alerted, but most hospitalists, especially in our area, ER doctors know to order a tick panel and also to alert the lab to look for the malaria-like forms, and then PCR DNA tests are positive. Treatment is another problem with Babesia. It's usually easily treated, but this has gone way too far where some doctors and patients think you have chronic babesiosis, and there's no real evidence that it lasts for more than 3 or 4 months. So I think I'll just stop there, and then there'll be time for questions.
Yes. Thank you very much. This brings us to our question and answer period. I would like Dr. Donta and Dr. Telford to please sit at the table. We will come around and collect your questions on the cards. Green were for Dr. Telford and yellow for Dr. Donta.
I thought about staying over. That's okay. It's an hour and a quarter. I live in Falmouth, so it's not. I'll just take my. I needed a GPS to get here, so I'll just.
It's tricky. Getting back to 495 is going to be a pain.
Well, I'm gonna put my GPS back on.
Yeah.
And see how to do that. So the reason the deer are such a target is because they bit down. It's just can't get on
No,
them.
it's more than the old tick has an affinity for deer.
How does it get on?
You come out tick collecting. Well, you don't even have to come tick collecting with me. You drag along the edge of the vegetation. They're sitting at the edge of the twigs with their legs out. And they hear and they smell.
Okay.
Now, this one's yours, though. Yeah.
Okay. Well, you better take. Well, we'll see. They'll come up. You can.
But I think it's more than that. I think that if people studied the skin secretions, things like that of deer, they would find them attractive to ticks. You can't get funding for it. It's called a fishing expedition.
You might mention something about that, and are the deer asymptomatically infected?
Yeah,
Or the
they
mice
are.
asymptomatically?
Well, somebody did try to attribute carditis in deer to Lyme spirochetes.
Because that's always an interesting question.
But
How come the deer aren't
there are
sick?
good in vitro studies that show that the deer complement lyses borrelia.
Right.
Complement. So, it's not likely.
Okay, let me look at these to see if there are combinations of things.
Well, let me triage the
Yeah, that's what we have to do.
questions, yeah, because we're not going to have hello, yeah. We're not going to have time for all of them.
No. You might address that one.
Material load introduced per tick. Now, if you survey in the ticks, the median is roughly 4,000 spirochetes.
Yeah, that's what I
But
said.
I've seen them as hot as 20.
Yeah.
Dose and severity of disease really needs to be looked at.
And we stand to reason that there is.
It's classic.
Yeah, I mean, it's a combination of susceptibility and inoculum.
Yeah.
Maybe we could alternate.
Sure.
Uh-huh. Just what I was asking.
This is supposed to be a joke. I suggest Sam get started.
We'll flip a coin.
Yeah.
No, you got
If
more
you're-
cards.
No, go ahead, start.
Okay.
And then
Well, I don't know whether we're going to get to all these questions, but perhaps Barbara and the Board of Health can find a way to post answers online somewhere for those that we don't address. I recently read a post indicating that larval ticks were found infected. Is it possible that the mother ticks pass on the bacteria to larvae? The answer is no. However, the companion infection, Borrelia miyamotoi, a relative of the Lyme disease bacterium, which we recently identified as capable of causing human infection, is inherited by ticks. So larvae are not necessarily harmless. It's just that it's probably pretty rare for someone to encounter a infected larva. So by and large, August and September are pretty harmless periods. That doesn't mean you shouldn't be using repellent or treating your clothing. Repellent, particularly because August and September are when mosquitoes are out and about, possibly carrying West Nile virus and Eastern equine encephalitis, and you don't want either of those either. So if you're going to be outside, it won't hurt you to use repellent. DEET has been 150 million doses of DEET used since the 1950s. There have been 24 adverse events. 24 out of 150 million. Do the math, is how risky that is.
one question here is, are there prospective clinical research studies taking place that are gathering data that will improve the understanding of Lyme immunology, leading to better biomarkers and diagnostics? And the answer is yes, there are some. We need more. John Alcott from Johns Hopkins is doing a nice progressive study collecting sera of patients with documented Lyme disease and seeing their immunologic and other responses with what's called proteomics and other newer methodologies. We should be able to come up with not just patterns, but markers that will eventually be helpful for physicians and for patients.
There's a question regarding Damonix tubes. Pros and cons of using Damonix tick tubes for control of deer ticks feeding on mice. Are these tubes harmful to domestic animals? The answer is no. The permethrin is bound very tightly to the cotton. And in fact, if you leave the cotton balls out in the sun, they're inactivated after 2 days. If you were to dump a case of Damonix in the wetlands, you might kill some fish or frogs. Cats are very sensitive to permethrin, which is why there aren't a lot of topical applications for cats. Nonetheless, the amount of permethrin on the cotton in these tubes is very, very low, and they're deemed harmless to cats, dogs, whatever you have around your yard.
There are questions here about what tests to do for Lyme disease, and there's another question about it. The general evaluation specifically focused on Lyme disease and co-infections is that aside from getting general blood work, that the Lyme-specific tests are what's called an ELISA screening test. And the CDC recommendation has remained that it should be a two-tiered test. That is, the screening test is negative, you don't proceed to the Western blot. I do take issue with that because in the patients, that works for the patients who have the big swollen joint, they're positive, and the Western blot is positive. But my own thinking, and it isn't just mine, but many others have realized that the Western blot, which is a more specific, because it details the immune responses to specific proteins of the bacteria, is a better way to analyze that possibility. And to me, it's not the numbers of bands, it's which ones. The 23 KD band, as it's called, is highly specific for the Lyme bacteria, as is the 39 and is the 83/93. So when I see those reactions, my suspicion that the person might have Lyme disease is heightened. There is another test called the CD57. It's an immune marker that's not specific for Lyme, but what needed to be done in the one abstract or short publication, we need longitudinal studies because the CD57 can be up and down depending whether you're sick with something else or not. But in general, patients may have low CD57 levels. You may also have a low vitamin D level, but then the general population might be low in D. Although I'm intrigued, the vitamin D from some mycobacteria TB studies might actually be like your body's natural antibiotic or anti-inflammatory. So I include a vitamin D test as well in those. Now, one question is Advanced Labs, which is a relatively new lab, is now claiming that they can culture the bacteria from patients with ongoing symptoms. And I think that they need a few more tests to validate their findings. The findings that were published were cross-sectional and really were not able, and they need to have blinded specimens submitted to them. And again, longitudinal pre and post-treatment to evaluate that. I did look at their culture methodology. Some of it sounds like maybe they're trying to be able to culture it more successfully, but they were dismissive of the PCR DNA correlation. They didn't do it, and their excuse was, well, it's not as sensitive. Well, there are ways to make the DNA more sensitive. And so I'm a little bit leery of it. I think it still needs to be shown. And from my model, I doubt that the bacteria are actively in the circulation. I may be wrong in chronic disease.
Can ticks enter a home on clothing, then transfer to another person in the house? And the answer is absolutely yes. If you are rolling around in the bushes and you take your clothes off or you sit next to someone in the easy chair, ticks could be crawling on your clothing and go right to that person who has never been outside. And that's why the Globe had a little article on a student in Braintree who did a science fair project. The CDC has long recommended having outdoor clothing and taking that off before entering the house, and if you must reuse it, put it in the dryer for an hour on high. It's overkill, but it'll certainly do the job, and what was novel about the Braintree Students Project is that it didn't take that long to kill them. Certainly putting your clothes, your outdoor clothing, in the dryer, changing your clothes will greatly help you. Putting your clothing in the dryer will help, but so will leaving it outside in on concrete in the hot sun. I would certainly be aware, though, of using the dryer if you have a lot of poison ivy around your home, because there are some people who have terrible poison ivy allergies.
Would you comment along that line about doesn't a washer, putting it in a washer with detergent, kill it?
Apparently not. The ticks, these things are pretty amazing in terms of how long they survive. In fact, the way I rear ticks in the lab is I have an animal cage and a moat of water, and I'm lazy sometimes. The ticks fall off the animal, crawl out of the cage, and get caught in the water, and I can leave them there for 2 weeks, and they're just fine. Surface tension will prevent them from sinking to the bottom and drowning. And so the detergent might help get rid of waxes on the outer surface of the tick, which will render them more susceptible to infection. But I've actually had live ticks come out of the wash. So it's the dryer. It's dry heat that is their enemy. It's not wetness. Wetness, there is a paradoxical effect. If a habitat is too wet, the ticks can't thrive because they're susceptible to fungal infections. That's my greatest enemy in the lab, is fungal infections that kill my ticks. So, but dryness, hot, dry is bad for ticks.
There's a question here about would I address Bartonella, and I'll ask Sam as well. My understanding is that it's not a proven organism that can be recovered from deer ticks. What's your understanding?
It's unclear to me why people are focusing on the deer tick as the vector of Bartonella, because you have the vector in your lap every night, and that's the house cat. The cat and its fleas are the maintenance host for Bartonella. We have plenty, it's what we call in epidemiology confounding biology. That is, you have ticks all around you, and you have Bartonella, which is truly a problem. It is a tremendous disease that can be difficult to manage. It doesn't mean that because you see ticks in your yard that they're transmitting it to you. But you're overlooking the real cause, which is your kitty cat, especially one that goes outdoors, and the fleas. You can easily find Bartonella in fleas by simply opening them up and smearing them on a piece of glass and staining them.
And this addresses the issue of overlapping symptoms with differing diseases. And the problem with Bartonellosis in humans, Bartonellosis is a problem for veterinarians, and testing is not always accurate. And what we have now are basically the same antibody tests for Bartonella. There is a PCR test too, but basically you have to rely on the symptoms. But I think that in the frustration of many physicians who care for patients with chronic symptoms, presumably chronic Lyme, the idea that they might have Bartonella comes from, well, they had this stripe, they had this red stripe, and isn't that a sign of Bartonella? I don't know what some of these nonspecific rashes that I see in so many patients with the chronic symptoms. Is it some kind of toxic, hypersensitive reaction? So I think that some of that may be overly diagnosed. But as far as treatment, standard treatments would include doxycycline or tetracycline. Some have used something called rifampin in addition to that. But it's sensitive to a number of organisms. It should be noted that even AIDS patients who get Bartonella seem to recover in the course of a few weeks of treatment. So I think one needs to be careful with presumptive Bartonella, let alone presumptive Lyme, how far you're going to go and treat patients. In general, these antibiotics are rather safe, but like with any other medicine and other profiles, you need to be careful about not overusing any particular medicine or antibiotic.
We shower, we check, how can you be sure a tick must be on for that length of time? The studies that demonstrated grace period go back to the 1920s with Rocky Mountain spotted fever. It's been seen multiple times with multiple different tick infections. The reason we know about Lyme disease and the grace period is because some poor schmo graduate student at Harvard had to sit up around the clock for 4 days pulling ticks off of animals that he had infested. So you take a bunch of hamsters, you put ticks on them, and at 12 hours you pull the ticks off and you see whether the hamsters become infected. At 24 hours you pull them off and see if they become infected. And 95% of the animals are protected at 12 or 24 hours, and then the rate goes down. That is, as time goes on, it becomes certainty that they become infected. That's how we know. It was done by experiment, and there's a biological basis for it.
I guess you can ask, though, is it reasonable to assume that that same dynamic takes place in humans? How well studied is that particular dynamic? Does it really take as long for a tick on a human or in a particular location, like an earlobe or another area that might be closer to where there is blood, for the organism to take that long? And a corollary to that is, if a tick is already engorged with blood, should you not make the assumption that the possibility of transmission has taken place?
Yeah, those are two good points. We will never know the answer for whether humans are more susceptible. However, I do note that we have received back criticisms of our tick feeding trials on people about, well, you know, we know this works on animals, but mice are way more sensitive than people are or primates are in terms of getting infected. So I think if anything, it's the other way around, that we use the most sensitive test, which is mice. They are exquisitely sensitive to being infected, whereas we don't know whether that's the case with humans. Now, every once in a while, you will pick up a tick that has already fed on something for a little bit, got indigestion, and fell off, and they're ready to go. And in fact, early on, people noted a correlation, an association with people who had Lyme disease and cat ownership as opposed to dog ownership. These were the very original studies done by Alan Steer epidemiologically in coastal Connecticut. And we've all sort of scratched our heads over years going, well, dogs, I mean, people get more exposed because they're walking the dog? Surely the association was wrong. It turns out that deer ticks don't like cats too much. It's an individual thing, but in general, they don't like cats too much. They'll get on a cat, they'll feed for a while, and then the cat will either groom it off or the tick will just detach spontaneously. So if the cat has fed the tick for 24 hours or more, and that tick crawls off in your bed because you like to sleep with your cat, yeah, you're going to get infected a lot faster than just 24 hours. So there are exceptions, and when I say there's a general grace period, it's a probability thing. It's much more likely you'll get infected if you let it feed for 24 or 48 hours. It's much less likely if you let it feed for less than 24 hours, to the point that it's almost an order of magnitude difference.
There's a question that addresses immunosuppressive medications and steroids, and do they adversely affect the process of Lyme? In general, infectious disease physicians are leery of any steroid use or immunosuppressive use during an active infection. But whether continuing steroid use will accelerate the Lyme infection is unknown. I think that when I get asked on a practical matter, I've got a hip that is arthritic, not from Lyme, but from some other guy, and my doctor says to have a steroid shot. I tell him that's fine. But if it's a series of more than a few steroid shots, or they're going to be on it, I tell them I'm not sure, but I'd rather you weren't on the immunosuppressive agent.
Why don't deer and mice get Lyme? Does any other animal? In the laboratory, we have mouse models, inbred mouse models of Lyme disease. We can reproduce the joint swelling. We can reproduce the heart problems. It's been hard to reproduce the neurological issues of late Lyme disease. But deer and the natural hosts, deer actually have a substance in their blood called complement. We all have complement. It's one of the main nonspecific effectors of immunity that we have. But every animal has its own kind of complement. Through evolution, they've evolved their own kinds of complement. Deer complement kills Lyme disease bacteria. So they don't get Lyme disease. Somebody did a study once showing that there was some inflammation in the hearts of deer exposed to Lyme, but we don't know whether there were other infections that might have caused that. In the laboratory, white-footed mice, if you have very young ones and you infect them, yes, they get carditis. There's inflammation in the heart muscles, but it goes away within 2 weeks, and they have never another effect. The reason for this, these animals have evolved over thousands, if not millions of years, a relationship with the Lyme disease bacterium. They've come to an agreement between them. They use each other to get around. What the benefit is of mice and deer to have Lyme disease around in the environment remains to be elaborated, but there's certainly a clear reason for Lyme not wanting to cause pathology in deer and mice.
There's a question here about biofilms, and this also then raises the question about cysts or cyst forms. There have been, with the cyst form, the experiments were in the test tube, in vitro, where one of the investigators from Norway put the bacteria in water. You put any organism in water and it'll curl up. Then he used metronidazole flagyl to treat that particular situation. And when he took those bacteria and tried to infect animals, he found it harder to infect the animal. That's kind of not the best rigorous research upon which to base a lot of concern. It's true that many bacteria, most bacteria, alter their shape when they get into certain tissues. And so I, as a practical matter, answer that kind of question. It's moot whether it's a cyst form or not. The more important point is it active or is it dormant? And if it's dormant, there isn't anything we can do about interfering with its metabolic process. So I think that I'm not fond of the experiments upon which they were based. There's another issue with metronidazole. That's a group of antibiotics. It turns out that there are no sequences in the genome of the bacteria to metabolize metronidazole. So any effective metronidazole has to be an indirect. And it's such a powerful agent on your intestinal bacteria that, and when people take metronidazole for other reasons, they generally note a big change in their intestinal flora. And I'm thinking that maybe what you do transiently, just as cholestyramine, Questran was being promoted to treat Lyme, what you're doing probably is detoxifying or eliminating some of the metabolic byproducts, which may be to the normal patient not causing any symptoms, but maybe a Lyme patient is hypersensitive because of the nervous system involvement, just to use a crude term. And so they feel the symptoms. This is the same as somebody saying sugars feed Lyme. There's no reason to think that that is happening, but nonetheless, these things come up because a patient noticed that when they eat more sugars, their symptoms flare. And that's true, but that doesn't mean it. So explanations come about. With the biofilm, there are usually wound infections in which there has been, I think, demonstration that it may be difficult for the antibiotic to get to the organism if the Staphylococcus usually produces the biofilm. And so this has been translocated to do the Lyme bacteria produce a biofilm. If so, maybe under the skin, but it's still going to be an issue. But intracellularly, there's no way you're going to get any biofilm buster inside the cells. So I think these are wishful thinkings, and I'm not sure how productive an approach is to focus on those particular issues.
I'm afraid we're reaching the end of our beautiful evening. Do you have one more question each of great significance, or are you sort of have reached the point where you say...
Well, there's no such thing as a bad question. They're
No,
all
never,
good.
never.
We'd love to sit and answer them all. One
Make a
last one
short
for
one.
me is, why are we relying on fair chase rules to reduce the deer numbers? Will the state ease up and allow longer seasons, more deer per hunter, and so forth? And the point is, it comes from the community. If the Board of Selectmen, under a mandate from the town, from the citizens, asked Mass Wildlife for help in increasing their take, Mass Wildlife would do whatever they could. They can alter local statutes, local regs. They did that for Nantucket. They allowed Nantucket to have a special hunt in February, a long time ago. This is 2005. It took us 3 years to get the town behind it. Selectmen asked, Fish and Wildlife came out, held a meeting, an open meeting, and modified their local regs to allow a February hunt. They took 200 animals out of that 1 week of hunting. Sadly, it was right after a huge snowstorm, and the middle of the island was impenetrable. And they had 600 hunters on Nantucket walking round and round and round, trespassing with hunter orange all over the place. Luckily, there were no accidents, and what most people don't realize is that in the dead of winter on Nantucket, they dropped $1 million worth of money in the hotels and restaurants and bars. So it was a real boon. It was a real boon for the island. Fish and Wildlife said that if they had another two such hunts, they would bring the deer population on a negative slope. So at the end of 10 years, they would actually come down to a very manageable level. Dog walkers and those whose properties were trespassed upon took up a petition drive and got 3,000 signatures and went to the selectmen and said, rescind this, and they did. But they will do it. They will consider requests to increase the take. But people have to realize that deer are a natural resource. The state manages them. They're stewards of our resources, and they want to see that the animals are used, the venison is used. In this day and age, with the economy being what it is, I'm surprised that communities aren't a little more aggressive about trying to take more venison and distribute it to the needy. It's a wonderful protein.
Well, I would like to say one thing in response to this, because we have tried and reached out to Mass Wildlife to change the duration of the hunting season and all sorts of small variations, even just the setback, as you very well know, and you can't get anywhere unless you have a real large mandate of individual. Small individual towns will never be able to do it. Nantucket is a different story, but I think we are beginning to run quite late. I want to thank you very, very much for coming, both of you, and I want to...
Thank you, Sam.
And I want to thank all of you for coming and asking such good questions. Thank you and good night.
Hey, good to see you, Dan. A lot of people have been talking about you in the last 2 days. Some folks on Nantucket heard about your business. Which one? Well, you're